welcome everybody to our last session of Reproducibility Basel for this semester. And it's my pleasure to welcome Fiona Ramage to you. She is a postdoc at the Camarades Research Group at the University of Edinburgh in Scotland. And in that research group, they do systematic review of data coming from animal experiments. Fiona has done her PhD in mouse behavioral studies, and now she is fully dedicated to meta-analysis and to promote the principles of reproducible science. And she is also active in the board of the reproducibility seminar in Edinburgh. And we are really looking forward to a presentation entitled Meta Research, a valuable tool to increase reproducibility in preclinical and other research. So Fiona, the stage is yours.
Okay. Yeah, thank you for the introduction, and thanks everyone for inviting me to talk a bit about my field of research. For you to be able to start to understand how I ended up in that field and how I shifted from lab studies, I wanted to talk about my experience a little bit in science. A fun fact about myself is that I was born in Basel and raised in the Alsace region nearby. My Swiss German is not up to scratch, so I won't even try. But yeah, I'm from nearby. I did, however, move to the UK to study a bachelor's in pharmacology and physiology in 2014. Well, I graduated in 2014. We were one of the only 3 undergraduate universities in the UK that encouraged us to do in vivo lab work. So I did mouse behavioral studies already as an undergrad. I did a master's in integrative neuroscience at the University of Edinburgh, again doing in vivo research. And in 2017, I started doing my PhD in nutritional neuroscience, where I was trying to understand how high-fat diets affected cognition in mice. What ended up happening is I did the first 2 years were behavioral diet studies, and then I ended up switching to meta-research. After finishing my PhD, I started doing a postdoc with Camarades, and I'm currently working on synthesizing animal research in psychiatry and involved in a lot of projects to improve reproducibility and research quality across the university and further afield. So why did I switch methods halfway through my PhD? What I started with was I was doing a replication study where I was trying to demonstrate that short-term high-fat diets impair cognition. If we got positive results, we were then going to look at how these short-term high-fat diets impair cognition, kind of hypothesizing that the mechanisms may be different than longer, more sustained changes to cognition. So what we ended up doing is we fed mice one of these 4 delicious diets. Either the blue one is high fat and sugar, the pink one was ketogenic, which is just high fat, and then there were 2 control diets. I did object recognition tasks, where mice were tasked with identifying a novel object in a novel place, or a more complicated task, identifying an object in a novel context or in a novel place and context. How these work is we'd expect if a mouse has intact memory and both objects are equally interesting to a mouse should always explore overall the novel one more than the familiar one. And if we do observe more novel exploration, that would indicate intact memory. Here are some figures from early on in my thesis. You can see that mice consuming a blue high-fat diet ended up having lower ability to discriminate the novel objects, performing below threshold, below chance, whereas the other 3 diets seemingly had intact memory function, not great, but better than the high-fat. We also got an independent scorer to score all these videos blinded, and we did a Pearson correlation looking at my original unblinded scores versus the independent blinding scores. And we saw that they had a strong positive correlation with an R coefficient of above 0.7, and this had a P of less than 0.0001. So we thought it was overall positive. But what I decided to do is I rescored all of these videos myself because I didn't really quite trust this. And when I scored them blinded, all of my results vanished. And as you can see, barely any mice are performing above threshold. Like none of even the control mice are able to discriminate a novel object overall. These are the 2 most complex tasks. They did the simpler task a bit better. And although all these scores were completely different, they were still highly correlated. Here's 3 sets of correlations between the 3 sets of scores, and despite strong correlations, the results were not the same. And why I thought this was an issue is blinding clearly has a huge impact on the study outcomes, with bias of the outcome assessor coming into play. It's particularly evident in subjective tests. I was having to look at whether a mouse was interacting with an object, and that often has quite fuzzy boundaries. And importantly, blinding is only really reported in about 10% of animal studies or animal behavioral studies, which was a bit worrying. While I was doing literature reviews, I also came across more problems in my field. There was poor reporting of other measures to reduce bias, such as randomization or reporting of animal dropouts or replacements. I found experimental design problems with issues such as pseudo-replication, no calculation of sample size, like statistical power, improper use of statistics, using the wrong controls, doing things like injecting massive amounts of glucose into mice and then doing behavior tests afterwards, which obviously would cause inflammation and potentially interfere with the animal tests, and not recording food intake to control whether anything was, any of the dietary effects were due to increased caloric consumption and weight gain rather than just the nature of the diets. There were so many issues that I came up with, and that made me think, can we even trust published literature? With all of this combined, as well as my own health issues, which made lab work more difficult, I decided to switch my tactic. I kept the exact same research question, and I thought, how can I address this, but with a different method, which is how that got me to design a systematic review and meta-analysis in preclinical research. And I'm going to introduce to you how it works. So when you have an in vivo study, typically you'll have a disease or a treatment group versus a control group, either without the disease or without the treatment or with a control treatment. And you'll assess a number of outcomes to determine the impact of the disease or treatment on this. This could be, metabolic outcomes, cognitive outcomes, that kind of thing. Typically, these kind of experiments are hypothesis testing using statistics, but they can also be observational or exploratory. Meta-research, in contrast, is research on research or research on research practices. You'll come up with a research question, identify all relevant evidence, analyze and summarize it using methods such as systematic review and/or meta-analysis, and then you would typically confirm or refute the findings of individual studies, and you would also get to explore sources of heterogeneity, why different studies show different results, and where bias might intervene. I want to give you a brief overview of the different steps involved in this kind of research. Typically, you would formulate a research question and design a search strategy where you would try and identify all relevant literature. You would design ideally hierarchical inclusion and exclusion criteria, deciding which studies you will retain and declaring this beforehand. You will then proceed with study selection and data extraction. You will need to also define data to collect, analysis plans, and how you plan to deal with bias. Typically, this will be done by registering a protocol beforehand, before data collection is started in a registry such as Prospero or OSF. You would then do your systematic search, usually covering several common databases and a comprehensive strategy, which is helpful to be designed based on previous studies or with the help of a librarian or a such information scientist. You would then do a screening of studies for inclusion to decide whether they're relevant. This would be done by 2 independent people and then reconciled by discussion with a third, ideally. You would then extract the data, both qualitative and quantitative, and again, reconcile it, and then proceed with analysis. To show you a bit what's involved in a meta-analysis, here's some dummy data that I created. The first thing you'd do is you'd choose an effect size measure depending on how your data are. There's mean difference, normalized mean difference, standardized mean difference, or where data is not continuous, there's other methods such as odds ratios. So you'll just determine these based on your data structure. If everything's measured on the same scale, mean difference may be fine. If you're measuring across completely different tests and different units and things like that, you might be looking at standardized or normalized mean difference. You then end up having complicated equations that you have to fit your disease and control values and errors and sample sizes into. You will then weight your effect sizes, either by sample size or inverse variance. You may nest effect sizes that are dependent, to combine several effect sizes into one. And then you would then calculate a summary effect size applying a meta-analytic model. You may have heard of fixed effects versus random effects. I'm not going to go into it, but essentially a fixed effects model is much simpler, which just accounts for within study variance, assuming that all measured effect sizes are just different measures of the same thing. Whereas random effects also accounts for between study variance, where we assume that, in fact, there's not one true effect size, but several different possible effect sizes. Typically, you would then draw something like a forest plot. This is not a particularly good one. This is just extracted from my thesis, and it's a bit cropped in. But you can see there's 0, which is 0 effect. You would have positive effects with a standardized mean difference, indicating that here a high-fat diet would improve cognition. So this is just the very bottom of the bar. Obviously, there were a lot more evidence that high-fat diet worsened cognition, but there wasn't space for it on this plot. And you have the mean effect estimate, you have the effect estimate for each study, as well as a 95% confidence interval. Another way to look at this data is to group the effect sizes per subgroup and calculate a pooled effect size per subgroup. Here, for example, we could see that grouping by sex, we could look at the effect size of high-fat diets on cognition in male animals, female animals, where the sex wasn't specified, and where the sex was mixed, using both male and female animals. And this was different. So that's just one way of looking at meta-analytic data. And then after that, you would look at heterogeneity in effect sizes and why these effect sizes are heterogeneous, which factors influence effect size. It's also worth mentioning that animal meta-analyses are much different than clinical ones, for which the methods were originally designed. The study design of a clinical trial is typically quite simple, whereas in animal studies, it can be very complex. Populations within a clinical study is relatively diverse, but relatively uniform between different clinical trials. Like everyone's a human being, typically of adult age, possibly with the same illness. Whereas in preclinical studies, it's very uniform within a study. For example, using a very specific genetic strain of animal, maybe even genetically related with, like from a single litter, in controlled conditions, but then very diverse between studies, with different studies using different species, different types of experimental designs and everything. Reporting for clinical trials is standardized, whereas reporting of preclinical studies is often poor and incomplete and varies a lot between studies. Data analysis in clinical trials is therefore straightforward, whereas in preclinical studies, doing a meta-analysis can be complex and really varies according to the type of data you have. Clinical trials tend to be, meta-analyses tend to be confirmatory, where we suspect the effect of a treatment and going to investigate that, and then determining how effective the treatment is. Whereas a meta-analysis of preclinical studies will be exploratory and will look at sources of heterogeneity, since there is not one true pooled effect size. Usually we'll be more interested in what causes effect sizes to vary. So this is the main part of my talk, which is I'm going to talk about how meta-research, specifically preclinical meta-research, can make research more reproducible as a whole. Firstly, meta-research itself usually uses reproducible and transparent methodology. They will be systematic, where you will systematically select and analyze evidence, and hopefully this will not be flexible. It'll be easy to report and easy to reproduce. It's done across multiple databases to capture all available evidence, which are then, and using comprehensive search strings, which are then reported when you write up the systematic review. You consciously limit bias as you're conducting a systematic review with multiple reviewers and then reconciliation of any disagreements. And you have to plan and declare the methodology beforehand. This leads on to the next point, which is systematic reviews and meta-analyses are typically pre-registered and can be peer-reviewed or at very least approved on registries beforehand. For example, I'm an administrator for Prospero for animals, where you review protocols of animal studies and confirm that they're passing quality checks. You can also pre-register your study on places like OSF. And systematic reviews and meta-analyses typically have to follow quite strict reporting guidelines. You may have heard of the PRISMA checklist and also come across something like a Prisma flow diagram, where you really explicitly state how you identified studies and how you determined whether they were relevant or not by including or excluding them at different stages. There's also loads of emphasis on data sharing, code sharing, and so on. And this is why overall, it's very commonly viewed that systematic reviews are right at the top of the pyramid of evidence as like the most valid form or the most convincing form of evidence, rather than single studies or, for example, a critical review that's not systematic. The second point I want to make is that meta-research often offers insights into research that single studies cannot provide. Single studies, particularly in animal studies, are small, and they may have very conflicting findings, at the very least due to just very different experiments. When you combine them using meta-research, you end up with a large combined sample size with maybe hundreds or thousands of animals rather than 10. And you get to look at the influence of the different factors that vary between studies on the measured effect size, like I mentioned earlier. There's several ways of doing this. You might look at the influence of experimental variables, such as animal species or age, or the drug dose that you give, or how the disease is modeled. And this is, again, the example from earlier, where we looked at the difference that gender makes, not gender, sex in animals, in mice and rats specifically, makes in the impact of high-fat diet and cognition, with male animals and where sex was not specified or where there was mixed studies having a greater effect than in female animals, for example. You may also be able to look at relationships between outcomes. So this is known as a meta-regression, and it's extracted from one of my colleagues' theses. It's a bit different than a correlation. It's more like how the variation in one variable, here on the x-axis, the reduction in amyloid plaque burden, can explain variation in another variable, such as reduction in tau pathology. Here, an R-squared value of 0.54 does suggest that they're related, and that one variable does explain variation in the other, which is not really possible in single studies. There's also an analysis of quality variables. For example, here we can see that animals that were randomized to group to their experimental or treatment group has a smaller effect size than where randomization was not reported. The third point I want to make is that meta-research, a key point is that meta-research doesn't only evaluate research itself, but it evaluates how research is conducted. We may look at the internal validity of studies. An example is looking at risk of bias. This is risk of bias across a large pool, about 250 studies in the field of high-fat diets and cognition, and following different criteria as laid out by the Circle's risk of bias tool, determining whether risk of bias is either low, where we're convinced that they've done a good job to limit bias, high, where we're convinced that there is a serious risk of bias, and then unclear, where essentially the reporting is just too bad for us to determine anything. We may look at study quality. So these were the ones from my thesis again, where there's many ways of doing this. You can customize it, but you can, I split it by whether I had low concerns, possible concerns, or significant concerns with the study quality, with issues such as unit of analysis, errors, or pseudo-replication, like I mentioned earlier, and no sample size calculation anywhere in the study. You may also look at reporting. So this is from a systematic review project that I'm involved in, the Galanos project, looking at the effects of a type of drug on psychosis in animal models, and using here a kind of expanded version of the ARRIVE guidelines, which is reporting for in vivo research. And typically what we do is we analyze per study whether this item was clearly reported, and then you're able to see trends across an overall field or subfield. Another thing which I don't have a figure for is evaluating the certainty of evidence. Based on the aforementioned criteria and other ones that you may come up with, you can do evidence evaluation and determine how confident you are that this evidence holds up, and then either upgrade or downgrade the importance of this evidence and how much it will contribute to your overall summary estimates. So you're able to kind of like weight the evidence according to how well the study is conducted. So we can look at all these factors. We can look at the impact on effect size. We can weight the evidence, and we can do sensitivity analyses. For example, we can exclude studies with a really high risk of bias and see if the conclusions change, for instance. Another thing that I'm going to mention later a bit more is that in meta-research, we also evaluate publication bias, which is the non-reporting of null or negative results, which unfortunately can lead to the overestimation of treatment efficacy. What looking at all of this does is it does encourage the adoption of better research practices, such as registering your study beforehand and trying to stick to your protocol, or at least declaring where you've departed from it. Publishing null findings, even if you don't find them necessarily that interesting, they are still important evidence if your study was well conducted. And also, we're trying to encourage the adoption of measures to reduce bias being incorporated into in vivo experiments. The whole reason that Camarades was founded was because my main supervisor, Professor Malcolm MacLeod, is a clinician who's particularly interested in stroke, and they were interested in why loads of potentially successful drug treatments in animals, about 1,000 of them, only led to one successful drug in the clinic, with everything else falling apart in clinical trials. So yeah, typically we design animal studies to inform clinical studies and to see whether a treatment is effective and whether it's safe before proceeding to human studies. Even if this treatment works well in animals, it doesn't mean it works in clinical trials, and this is called translational failure. It's also translational science, which is my field, is also kind of seen as a valley of death, where everything goes quite well in basic science, in the lab, and so on. Everything can go quite well in clinical trials, and where everything falls apart is in translational science, where you try to move from this bench research into humans. There's many possible reasons for translational failure. I've only outlined a couple of them here. Maybe the findings in animals are not generalizable to human health. Maybe, mice's bodies are different, and what we find in mice isn't applicable to humans. We also may not understand the underlying disease, but it could also be a quality issue. Maybe the preclinical studies are not reproducible. Maybe they use poor models for what they're trying to test, and maybe they are biased. So what meta-research of preclinical studies then does is we can examine these animal studies, see where things are potentially going wrong, just understand the evidence a bit better, understand the influencing factors that will determine how well treatment works, and we'll be able to evaluate the quality and bias of these studies. We can gain new insights into how everything works, both in animals and in humans, come up with new hypotheses and design new experiments to test these, and we can ultimately inform clinical trial design as well, and hopefully lead to successful translation from animal work into clinical studies. The final point for the benefits of meta-research that I wanted to touch on was how meta-research can limit research waste. If we think about, if we have a research question, a lab scientist would maybe immediately think about generating new experimental data. How can we design another small study that will test our hypotheses? Whereas doing meta-research would try and gain new insights from existing data, from data that's already published. If we design a new study that we don't really necessarily need, this could result in unnecessary duplication or replication of this study, whereas in meta-analysis, we can just reuse existing data. If we do this unnecessary study, like duplication of effort, or like design a study that doesn't really add to the knowledge base, this would waste resources such as researcher time, funding, money, and also animal lives if you're doing animal research. Whereas meta-research kind of does the opposite. It kind of takes knowledge that was generated from animals, from existing funding and people who had invested time to create data sets, and it really maximizes this usefulness and allows it to be reused. Meta-research is relatively resource light. I'd say the only one that really counts is time. Also, animal studies have to be just by design quite small, and you'll get quite a small part of the picture, whereas meta research, you can really get out an overview of the whole subfield if you do it right. If you design if you design a new experiment, you might obtain conflicting results which don't align with what you had previously. Whereas meta research, you may instead identify knowledge gaps a bit easier. New studies are vulnerable to bias, whereas if we do meta research correctly, we can minimize bias and evaluate the presence of bias and its impact as well. So overall, meta research, when well conducted and when compared to maybe an unnecessary preclinical study, can be less wasteful. I did also, the last part of my talk that I wanted to touch on was some challenges that we face in preclinical meta research, because I feel I've painted quite a positive picture so far, but I want to emphasize that it's not easy and there are lots of things that make this very challenging for us. A big one that my group is focusing on is keeping up with the pace of published literature. Considering I'm doing a series of projects on psychiatric illness in animal models, if I just did a PubMed search using very basic search terms, psychosis or depression or anxiety and animal, obviously not comprehensive, this alone led to 157,000 potential studies, most of which will be irrelevant, admittedly, but I'd still have to sort through them manually, with tens of thousands published every year, which becomes very hard to deal with. It's impossible for any scientist in their field to keep up with literature enough to do a literature review or be aware of everything that's going on in their field. But with meta researchers, it's hard as well. Even if we're staying at quite a surface level and trying to filter through the information using proper processes, it still overwhelms us. And the amount of research published just forever increases. And as I mentioned before, synthesizing research is resource intensive. When you consider that we manually extract data points from graphs and write down everything that's been done to an animal, and sometimes have to answer a form with maybe 100 questions per paper to get all the data we want to extract from it. So possible solutions that my group have developed is incorporating automated methods into this. We can do automated citation retrieval via API queries, where we will kind of go in the back end of these, for example, PubMed or Scopus of Web of Science, and automatically capture all of the citations published within the last week and kind of refresh this every week and just import them into our databases manually, avoiding having to do what I just did with the search from all time with, yeah. And what we then do is we still end up with really large amounts of citations. Maybe I think for depression, we had maybe 80,000 potentially relevant citations in animals, which is too many. So what we can do with machine learning is we get human decisions and we make maybe 2,000 or 3,000 human decisions of either include or exclude. And then we train a machine learning algorithm on these decisions and teach it to recognize features that would be present in a study that we would want to include versus features of a study that we typically wouldn't want to include. There's lots of things that can go wrong with this, but we do keep a close eye on the sensitivity and specificity of this approach, and it does seem to add a lot of value and save a lot of researcher time. And we then use text mining for feature extraction to extract, for example, which animal models were used, which treatments were used, and which outcomes were evaluated in these animals. This is just an overview taken from my colleague Caitlin Hare's AD Souls, which summarizes Alzheimer's disease research. This is a summary of all experimental models that have been automatically tagged in the Alzheimer's disease research that was determined to be relevant by the machine learning algorithm. As you can see, it's all kind of categorized into these hierarchical categories. So we get a broad overview of what is present in the database, which might identify knowledge gaps, but also allows us to go into these. And for example, if we're only interested in a certain model, it would allow us to go in and extract and subset those papers so we can just retrieve those ones. Another challenge is that, like I touched on earlier, in vivo research is often poorly reported. The minimum we expect for extracting numeric data is for them to be a measure of mean or variance and sample size, which is necessary to plug into the equations that I showed earlier. However, most of the time, well, not most of the time, some of the time, authors don't specify what the variance represents, whether it's standard deviation or standard error of the mean. So we have to kind of try and take the conservative estimate, but it's not ideal. And sometimes people will just report sample size as a range or not report it at all. There's often not clear detail on the timing and frequency of procedures used, or animal characteristics such as species, age, strain, or how outcomes were assessed. For example, which behavioral task was used or how exactly we measured outcome. And there's also poor reporting of whether the authors used measures to reduce the risk of bias, such as blinding or randomization. This means that, well, a meta-analysis of poorly reported research will be a meta-analysis with lots of gaps in it. Like, we won't be able to assess it properly unless it's properly reported. This is an example, again, from the systematic review of a type of drug treatment on psychosis. And you can see that for all these studies, everything was yellow, basically marked as unclear. Like, we were unable to determine the risk of bias. Typically, for these types of things, we tend to be conservative and assume that it probably, if it wasn't reported, it probably wasn't done. But again, it's not ideal. What are potential solutions to this? What we do is just encourage good reporting standards in vivo research, for example, following the ARRIVE guidelines. And we kind of hope that journals would enforce this a bit more as well, to encourage authors to, well, to, for example, make it mandatory rather than just optional. And the other difficulty, which I touched on earlier, is publication bias. If we test a hypothesis, typically we will have some positive findings and maybe some null findings. However, the null findings are typically seen as less interesting, less novel, less career-defining, and less impactful in journals, for instance. So then we tend to kind of select for only the positive and really interesting findings are kept. But when we do a meta-analysis of studies, this means that, for example, if 100 studies were conducted and only 50% of them had positive findings, we're then summarizing only the 50 positive findings and completely disregarding the ones with null findings. So we're completely overestimating the treatment effect. We're more likely to publish positive findings, like I said. It's also more likely that we would publish large studies, regardless of findings, than the small studies, just because of the amount of effort and funding that went into them. And also, typically, large studies measuring a lot of different things and us being able to publish at least some of them. But then what's missing is the small studies with negative findings. The reasons for publication bias, there's probably many of them. I can name two. That is research culture, people believing, the kind of publish or perish mentality, and wanting to have your results be as interesting as possible rather than just good science, which is often under-prioritized. And part of the issue is journals as well, like journals maybe not accepting null results as often as they accept positive findings. Potential solutions are estimating publication bias and its impacts, which we can do via statistical methods and meta-analysis. We can find where there are missing studies, and we might be able to even impute possible effect sizes that are missing from the data that would be present if all studies were published. We'd be able to hopefully provide more incentives for people to publish their null findings and again change journal policies. And another thing that could be beneficial is using registries for studies so if you've published a protocol, you're probably more likely to publish the completed study afterwards. Something else that's challenging is identifying all relevant evidence that we could use, because if we're doing a systematic review, it's systematic, we want to capture absolutely everything, and that's often difficult. The first problem is that in literature databases such as PubMed, you're typically searching by what's in the fields that are indexed in PubMed, which will be title, abstract, keywords, and things like that. But sometimes some things such as an outcome used, maybe you're only interested in open field tests, that won't feature in the abstract and it'll only feature in the full text. This is a problem that we're trying to tackle again with the automated citation, the automated synthesis methods. And yeah, we also want to capture like studies published at any date and eliminate any language restrictions and include grey literature as well to make sure we're capturing as much as possible. Another thing that's a challenge for us to identify relevant evidence is things such as papers that are behind a paywall and data and code that is not accessible. This may be solved by publishing open access, by maybe publishing your data as a preprint, and hopefully also sharing data and code, which in addition to giving us all we need, it would also save a lot of time if we had fully available data sets. A third problem with identifying all relevant evidence is when do we decide when we've reviewed enough? So I'll introduce the topic of living systematic reviews. These are systematic reviews that are continually updated, well, not continually, but periodically updated to incorporate any new evidence. They're particularly important when it's a fast developing field and new evidence is emerging rapidly, or when the review is really important for decision making, such as will a drug be progressed to clinical trials, in which case we really want to have all available evidence. So what we want to know is, do the conclusions actually change when you incorporate this evidence? And we're kind of deciding that if the field has stopped evolving so rapidly, or if every study seems to say the same thing and be in line with their original findings, maybe this is an area where we can maybe spare some effort, and maybe keeping this review in living mode isn't necessary. Finally, my last point is, I think it's actually my penultimate point, is analysis challenges that we face uniquely in animal studies and not in clinical studies. In the clinic, we would typically find one study with one group of patients and a second study with a second group of patients, and these studies are more or less completely independent. That means that the statistical assumptions outlined by the meta-analysis methods are met, and also these methods were designed for randomized clinical trials, so that makes sense. But in animals, there's a lot of non-independence of effect sizes and also non-independence of errors, which we have to account for statistically. We might have 5 studies. We might have 2 different authors, and then we might have 3 studies that are authored by the same lead author, by the same team. And logically, these should probably be more closely related than, for example, a study published by author 2 or author 5 would be related to author 1. Animal studies may also share animal strains. Maybe C57BL6 mice will be used by 2 strains, 2 studies, and maybe something like Wistar rats would be used by other 3, because the mice of the same strain are genetically related. Obviously, the effect sizes and errors should be more closely related to each other than they would be to anything else. This violates statistical assumptions laid out by meta-analysis methods, and leads to us overestimating, again, effect sizes. There's many other reasons for non-independence, which we tried to account for, such as multiple effect sizes within a study when there's multiple experiments that are done, reusing animals and measuring several outcomes in the same animals, maybe the same laboratory conditions, such as housing all the animals from different studies in the same animal room. The source of animals, maybe animals that are all bred in Charles River are more closely related than animals that are not, that are bred elsewhere, and also just experimental design relatedness. There's kind of 3 levels of solutions I've proposed here. If animals are reused, we may just do simple statistical things such as a sample size correction, dividing a control group by the number of comparisons that it has participated in, so that animals don't contribute to more than one effect size. We might also combine effect sizes that are related into one, such as essentially averaging them, but then we lose a lot of granularity in the data. We may also finally use a multi-level model, which is the ideal version, which is when you kind of combine the data statistically in several hierarchical levels, and you indicate when studies would present with a certain relatedness due to a shared animal strain or shared author group or anything like that. But all of these can only be accounted for if it's properly reported in the paper. Finally, there's also poor quality within meta-research. Like I said, systematic reviews and meta-analyses are typically considered at the top of the evidence pyramid, but if they're done poorly, that's not the case. They could be done poorly if the research question is poorly defined. There could be a poor methodology used or a lack of expertise from the reviewer team. Maybe we would introduce meta-bias or reviewer bias as we're conducting the systematic review, and then we might make the review non-reproducible or non-systematic, or report the review poorly, or have a lack of transparency in reporting. The only solutions I can really offer for this are training and education for the reviewer team. For example, my colleagues in Kameradies Berlin have a communities for open research synthesis where they provide training to aspiring meta-researchers. And there's also many tools that exist specifically for the design, conduct, and reporting of meta-research, both clinical and preclinical, which I'd recommend using. For example, registering your protocol in Prospero, adhering to PRISMA guidelines for reporting, and using really specific data extraction platforms, such as our custom one, Surf, which really allows you to kind of collect things in a more reproducible and transparent way. So where did I end up after all of this? I ultimately completed my PhD in I defended it in December 2022, and I was awarded it. My blinding study, interestingly, well, I wasn't able to publish my PhD data, but we did then decide that this was of importance. So we took a group of about 65 undergraduate students and made some of them blinded, analyzing my exact same videos that I recorded for my thesis. And then we looked at whether students, when they were blinded, would essentially view the object recognition task results differently, whether non-blinded students would maybe overestimate the effect of diet or the performance of mice in object recognition tasks. I was also delighted to be awarded a credibility prize by the British Neuroscience Association due to my kind of, I guess, mea culpa approach, where I decided, like, okay, I cannot publish my data and I won't worry about it, but what I want to do now is I kind of want to turn this around, and I want to do good science. Like, I don't want to continue trying to reproduce this study that I don't believe in, and I kind of lost a bit of faith in all the methods, but I switched to this meta research. I did a lot of research into how to do it properly, and then I proceeded to develop something of a passion for reproducibility and open science, and have tried to sort of share this message with other people at my old university and my current one. I founded reproducibility at the University of Dundee and ran it for a year and a half. And I'm currently leading up to taking over reproducibility at the University of Edinburgh. And finally, we're also creating a bunch of resources that will allow the adoption of good research practices across the university. This is a tool that we're designing for PhD students. I just want to thank everyone who's been involved in all of this work. This would include my team Kameradies mainly, especially with my supervisor, Professor Malcolm MacLeod, also my old university, University of Dundee, and the people currently funding my salary, which are welcome via the Galanos project. Thank you very much for listening to me, and I'll happily take any questions or participate in any discussion. Thank you.